Good evening, everyone. I am Vipul Delwal, Department of Pharmaceutics and Pharmaceutical Technology, Formulation, Naipur Mohali. Today, I will discuss about the oral delivery approaches of protein and peptide. Most of pharmaceutical companies in recent times are working on the approaches to deliver protein and peptide orally because there is many problems to deliver peptides and protein via injectable routes. So they are currently moving to the oral delivery approaches, protein and peptide. So in this presentation, we will discuss complete approaches and the recent technologies to deliver protein and peptide orally. So this is my flow of presentation. First, we understand the introduction and current market scenario of protein and peptide, then the significance and drawbacks of peptide therapeutics as compared to the biologics and small molecules. Then challenges to deliver protein and peptide. PPD stands for the protein peptide drug delivery, and the strategies of oral protein and peptide drug delivery. Then the chemical modifications. What chemical modifications can affect the viability of the protein and peptide? Then the sal sodium. This is the most widely used permeation enhancer in the delivery of protein and peptide orally. The use of sal sodium is called elegant technology. This technology used in the ribolysis formulation, which is the outbreak of oral delivery of protein and outbreak of oral delivery of GLP-1 analog, which is a peptide in nature. The absorption enhancement of peptide through sal sodium. Fatty acids, sodium caprate is absorption enhancer. Then the peptide in oral R&D. One more slide which I add in between this is the hydrogel formulation, which is the recent advancement to deliver orally protein and peptide. Then the conclusion. So in this slide we are seeing the introduction of our presentation. Has this bar chart depicted that the 45% of the marketed formulation are come under the intravenous formulations, and the 33% are the subcutaneous formulations, and the 14% are the intramuscular formulation, and only 4% are the marketed formulations which is orally delivered, and the other are 1% like inhalational formulation. In inhalational formulation there is a marketed formulation of insulin is called Afrezza, which deliver the insulin by a dry powder in form of dry powder inhaler. Okay, so why companies are focusing on the other routes? The reason is that the some issue facing by the pharmaceutical companies, and the some issue facing by the patients also, because the oral formulations and nasal formulations the high patient compliance as compared to the injectable formulation. So in case of intravenous administration, intramuscular administration, and the other is the subcutaneous administration. So in other administrations like intravenous, intramuscular, and subcutaneous, there are problem patient facing is that the lipohypertrophy and lipoatrophy. What is lipohypertrophy and lipoatrophy? There is the deposition and degradation of the fat at the injection site, which is the major concern in case of the subcutaneous and the intramuscular injections. So due to these reasons and the some other reasons which is facing by the pharmaceutical industries, they are focusing on the oral delivery. Okay. Here we discuss about the significance and drawbacks of the peptide therapeutics. The first, advantages and drawbacks of peptide over small molecules. As we know, there is some advantages like high specificity. In case of protein and peptides, there less of target activity as compared to the small molecule, and we get good efficiency and good safety as compared to the small molecules. But there is some drawbacks also in case of peptides and protein. So first is the low stability. As we know, the peptides has low stable than the small molecule because we need to maintain their 3D structure in their three-dimensional. Like there are secondary bonds which maintain their structure and efficiency, and therapeutic activity responsible for the therapeutic activity. And the other drawback is the short half-life. And the third one is low membrane permeability. It is the major concern in case of peptides and protein. Here I mentioned peptides, but this drawbacks also for the proteins. And when we compared peptide over biologics, so there is some advantage. So peptides and protein also comes under the biologics. So here the biologics means the molecular antibodies, monoclonal antibodies and all. So when we compare the monoclonal antibodies and the other high molecular weight biologics, peptides have some advantages like low immunogenicity, membrane permeability. So the peptides has a more membrane permeability than the other biologics, and the low cost, and the drawback is the short half-life and low stability. So now we will discuss about the challenges of oral protein and peptide delivery. The first challenge is that effect of enzymatic degradation. As we know, in stomach there is a pepsin enzyme. This pepsin enzyme is responsible for the degradation of protein into the polypeptides and the polypeptides into the subsequent amino acids. These amino acids will not show therapeutic activity. So this harsh environment can degrade our therapeutic peptides into the stomach. In intestine there are three enzymes: chymotrypsin, trypsin, and elastase, which degrade our peptide and protein into the intestine. And this enzymatic degradation causes their denaturation and degradation of our therapeutic peptide and protein. Then the physicochemical properties of peptide. The physicochemical properties mostly affect the viability of peptide and protein. The first one is the molecular weight. So as we know that the molecular weight of the peptide is more than 4 kilodalton to many kilodaltons. This high molecular weight opposes the Lipinski rule. So due to the opposing the Lipinski rule, they cannot cross the biological membrane. Another physicochemical property is the molecular charge. So they possess molecular charge into the stomach and intestine. And as we know that non-ionic species more readily cross the biological membrane as compared to the charge moiety. So another barrier is the intestinal epithelial barrier. So I need to hide myself. Okay, there is no option. So there is in intestinal epithelial barrier. The most affecting barrier is the role of P glycoprotein. The P glycoprotein is the causing the efflux of our peptide and protein and prevent the absorption of the peptide and protein through the intestine. And another effect is the CYP450 enzyme. This enzyme, sorry. So there is some extent of CYP450 enzyme present in the intestine, and this enzyme degrades the protein and peptide into our enterocytes. Then the other intestinal epithelial barrier is the tight junction of the cells. This tight junction prevent the entry of the biologics into the blood stream. Then the another challenge is to the mechanical degradation in GI tract. The mechanical degradation GI tract is due to the harsh environment on the GI tract cause mechanical degradation and denaturation or therapeutics. Then the effect of GI pH. So how GI pH can affect our peptide and protein into the stomach and intestines. So as we know that for activation of pepsin, there are acidic environment required for the activation. So this is our pepsin. And acidic pH, when pepsinogen is converted into the pepsin in acidic pH. Without acidic pH, pepsinogen cannot convert it into pepsin, and this pepsin is responsible for the degradation of peptide into the stomach. But as we know, the pH is increase in intestine. This increased pH is responsible for the activation of chymotrypsin, trypsin, and elastase. These enzymes are activated at intestinal pH. So when we one of approach which we can acquire, like decrease the intestinal pH also, and this decrease in intestinal pH, this decrease in intestinal pH can deactivate that enzymes and prevent the activation of these enzymes. So this enzyme is responsible for the degradation of our peptide. So this is the reason why GI pH can affect the our oral delivery of peptide. Okay, now next. So in this slide we will discuss about the strategies of oral protein and peptide delivery. The first is the chemical modification. So chemical modification, the process is called lipidization, cyclization, pegylation, and product. This we will discuss in the subsequent slides. Now we focus on the effective agents. So first effective agents is absorption enhancer. As we already mentioned, the ribellious formulation use the salcaprosate sodium, which is also a absorption enhancer. The other absorption enhancers is, one is the sodium caprylate, sodium caprate, sodium dodecyl sulfate. There are multiple absorption enhancers which can deliver the peptide and protein orally and increase the permeation through the maybe a opening the tight junction, maybe transcellular permeability. So the absorption enhancer most used in the pharmaceutical industry to increase the oral delivery of peptide and proteins. Now the enzyme inhibitors. There are some enzyme inhibitors which are used in the oral formulations. One is the soybean trypsin inhibitor. This soybean trypsin inhibitor increase the protein and peptide viability by decreasing the activity of enzymes like trypsin, chymotrypsin, and the other enzyme like pepsin. So the one example is the soybean trypsin inhibitor. Then the other is the modulation of the pH. So modulation, as already we discussed, the modulation of pH, how much it is important. In intestine, the degradation of peptide is caused by the trypsin and chymotrypsin. They activated in the particular intestinal pH. What if we decrease that pH by use of citric acid? Citric acid is decrease the intestinal pH slightly, and by the decreasing of the dead pH, the activity of the enzyme will reduce. And also we can done in the stomach also. And the other is the use of the mucolytic agents. The mucolytic agents is the example is the polyethylene glycol and the trimethyl chitosan. They bind to the disulfide bond of the mucus and they degrade the disulfide bond of the mucus and increase the permeability of the protein and peptide. The most important strategies is combinational strategies. So first is the transit permeation enhancer technology. This technology developed for the delivering of the oral delivery of the octreotide. They make the octreotide suspension by the use of permeation enhancer called sodium caprylate. And we depicted sodium caprylate by C8 because it contains the 8 carbon chain. And by use of sodium caprylate, they enhance the permeation by opening the tight junction. And the other method, formulation technology, they use polysorbate 80. The polysorbate 80 is increase the transit time of the octreotide into the intestine by the binding the mucous membrane. The another technology is peptides technology. Peptides technology, the focus on two phenomena. First is the modulation of the pH, and the second is the enhance the permeation of the pH. By the modulating, they prevent the enzymatic degradation, and by use of permeation enhancer, they increase the permeability also. Next is the GIPET technology. In this technology, the full form of the technology is the gastrointestinal permeation enhancement technology. So in this technology, the use of medium chain fatty acids. The use of medium chain fatty acid called gastrointestinal permeation enhancement technology. Medium chain fatty acid is sodium capridine, this sodium caprylate. So they are the two main permeation enhancer comes under the gastrointestinal permeation enhancement technology. The other one is the thiometrics technology. So what is thiometrics technology? In this technology we use thiomas, thiolated polymers. That's all. Now we will discuss the drug carrier system. So there are the delivery through the liposome. As we know, in liposome we can deliver both hydrophilic molecule and the lipophilic molecule. So according to BCS classification, our protein and peptide comes under the BCS classification 3 and 4. So they are the low permeability, but also in BCS class 4 low solubility. So some peptides and protein has a high solubility also. So delivering to the liposome can prevent their enzymatic degradation, plus can they increase their permeation. If we attach PEG molecule with our liposome, can also prevent by recognize RES system. So the liposome is the one of important technology or strategy to deliver peptide orally. Then the microemulsion, by using the surfactant and co-surfactant, we can prevent degradation and also increase the solubility if our peptide belongs from the BCS class 4. Then the another important strategy is use of hydrogels. The use of hydrogels are the substance which usually inert, but when they come in the contact of the intestinal fluid, they become swollen, prevent the degradation of peptide into the intestine, and also they increase the permeability. And the other method is the use of ionic liquids. The use of ionic liquids, the first of all we understand the what is the ionic liquids. So ionic liquids are the low melting point salts. Their melting points below 100 degree Celsius, so they can use to the deliver peptide orally. And the another mechanism they showed is that they remove the surface water. They remove the surface water from enzymes. So what removal of surface water can helpful for deliver peptide orally? So by the removing of the surface water, the enzyme will not show their action, like they degrade the, like they break the secondary bonds of the peptide, so they will not degrade by the stripping of water from the surface. So in this slide we will discuss about the chemical modification. So first, in chemical modification we will discuss cyclization. So how cyclized peptide has more viability than the linear peptide? The reason is that first is the no free N terminus and no free C terminus. If there is no free N terminus and no free C terminus, there is no cleavage sites for the peptide, for the intestinal enzymes and stomach enzymes. And the reduced flexibility, they increase the flexibility also, and which also was restricted peptidase activity. And as we know, there is a Lipinski rule, and Lipinski rule says that the high molecular weight compounds cannot permeate the plasma membrane, as we know in case of linear peptides. But in cyclized peptide, they are crossing the biological membrane. The reason is that they show chameleon characteristics. Due to this chameleon characteristics, they opposing the Lipinski rule. How they opposing the Lipinski rule? This is the question. The reason is that due to the chameleon characteristic, they exposing their hydrophilic groups into the aqueous environment, and in case of hydrophobic environment, they expose their hydrophobic group. It will increase their solubility also and permeability also. So their peptides and protein belong to the BCS class 3rd and 4th, so they have low permeability, and due to the chameleon characteristic, they have high permeability. This is the reason why cyclized peptide has more viability and more solubility than the linear peptides. Now we focus on the lipidation. Lipidation is nothing but attaching the lipid moiety to the peptide. So due to the attaching lipid moiety, they have low permeability, but after attaching the lipid moiety, they increase the permeability their because their hydrophobic nature is increased due to the attaching lipid moiety. So two types of lipidization mentioned here. First one is the covalent lipidization and the second one is the non-covalent lipidization. When we attach the lipid moiety by a covalent bond, we call covalent lipidization. When we attach non-covalent lipid moiety to the non-covalent bond, we call non-covalent lipidization. The another one is important is pegylation. So pegylation is also nothing but attaching a PEG moiety. This PEG moiety increase their circulation time, increase their permeability by restricted the restricted the activity of enzyme like chymotrypsin, trypsin, because they create a hindrance around the peptide. So enzymes cannot attack on the peptide and prevent their degradation through intestinal enzymes, and also reduce the renal clearance because their increased size also by the attaching the PG moiety. And the another one is the peptide change the peptide analog. This is the used least in chemical modification because it may cause toxicity and all, because we changing the D form to L form to D form, and by the changing of L form to D form we can reduce the enzymatic activity through trypsin and chymotrypsin. That's all in this slide. So from this slide we are focused on the permeation enhancers used in the delivery of oral protein and peptide. So there are multiple clinical trials are going on the use of permeation enhancer in delivery of biologics and protein and peptide. So as we know, the use of permeation enhancer can increase more efficiently by the use of other methods. So first is the mode of action of permeation enhancers. The first is the paracellular permeation enhancer, second is the transcellular permeation enhancer. So now what is paracellular permeation enhancers? So paracellular permeation enhancer is the enhancing the permeation of the molecules via opening the tight junctions, like first generation permeation enhancers, second generation permeation enhancers. So in first generation permeation enhancer, example is EDTA, oleic acid. So they not directly open the tight junction, but they affect the cell signaling pathway. By affecting the cell signaling pathway, they increase the permeation by opening the tight junction. This phenomena we will discuss when we discuss about the sodium caprate mechanism. So this phenomena will follow in this sodium caprate action also. And in the second generation permeation enhancer is the zonula occludent toxin. They are not affecting the cell signaling, but they are directly open the tight junction. So they are more toxic than the first generation permeation enhancers. The second one is the transcellular permeation enhancers. In transcellular permeation enhancers, they are directly increase the fluidization of the membrane, like salcaprosate sodium in ribolaceous formulation, and the other is the sodium dodecyl sulfate. So they directly increase the permeation and they are opening the tight junction. That's the difference between the transcellular and paracellular. So most of the permeation enhancers are the paracellular in nature, and they open reversibly to the tight junction. If they are not doing reversibly, so they may cause toxicity and they remove from the development pipeline. So in this slide we are seeing the types of permeation enhancer. Either here depicted the multiple permeation enhancer which can be used for the delivery of protein and peptide, and This permeation enhancers is mostly used by the development, and the multiple patents are mentioning this permeation enhancers. Like first one is the surfactant like sodium dodecyl sulfate, then the fatty acid sodium caprate, then the long chain fatty acid like lauryl carotene chloride, and the cationic polymers also chitosan salts, the chelating agents like EDTA, the bile salts like sodium trocholate. There are the multiple permeation enhancer which can increase like fatty acid sodium caprate, which increase the permeation by the increasing the membrane fluidity also, plus also affecting the cell signaling pathway. Affecting the membrane fluidity by the sodium caprate is less, but the affecting the cell signaling pathway, by the affecting the cell signaling pathway they open the tight junction, and this activity is mostly depicted by the sodium caprate. The another is the long chain fatty acid man laurethine creatinine chloride. This laurethine creatinine chloride affect the membrane fluidity and clouding modulation. The clouding is the tight junctional protein which is also affected by the sodium caprate. So now one is the cationic polymer is a chitosan salt, then the chelating agents like EDTA. So they complexation of calcium and magnesium tight junction opening. So by they binding the calcium and magnesium into the form chelate and increase the retention time plus permeation also. Then the bile salts like sodium trocholate, they increase the membrane fluidity plus tight junction modulation. So tight junction modulation they are primarily action. So in this slide we are focusing on the sarcoiprosate sodium, which is the part of the elegant technology, and another is the CNAC, and also we call this to snack. The most common name of sarcoiprosate sodium is snack. As many people already know its name, because it is the mostly used in the delivery of protein and peptide, and the well-known formulation called tribalases use this snack to deliver their semaglutide orally. So in this slide we are mainly focused the understanding the general mechanism of peptide and deliver the snack deliver the peptide. So this picture depicted the peptide molecule, and this picture depicted the snack molecule. When this peptide and snack, this snack will help to cross the biological membrane to this peptide, and after the crossing the biological membrane it dissociate from the peptide. But some studies also showed that some amount of snack also available near to the peptide after the crossing the biological membrane by the use of this snack or other excipients. So in ribalaceous formulation the viability of is still 1%, but the in after this 1% viability it shows the high amount of cells in the market, because high patient compliance is attached to this formulation. So it is showing the general mechanism. The deep mechanism we will discuss into the next slides and subsequent slides. This is the structure of sarcoiprosate sodium, and this sodium helps to create the basic environment. And the most important aspect of ribalaceous formulation is that they deliver the formulation in stomach. So in stomach is acidic environment, and this acidic environment dissociate our peptide. So by the use of single excipient called sarcoiprosate sodium, they increase the pH and increase the fluidization. So it is the most widely used excipient in current industry. So this, as we discussed about this sodium ion, so this sodium ion creates the little basic environment around the pH, around the our peptide. So it increases the microenvironment pH of the peptide, and this microenvironment pH prevent the acidic degradation and pepsin degradation. So in this slide we are focusing on the absorption enhancement of peptide through snack. As we already discussed one of the mechanism of the snack in semaglutide. So here is the peptide is our semaglutide. Consider this peptide is a semaglutide, because due to some reasons I did not mention the name of the semaglutide, I just mentioned the peptide. So in ribalaceous So in ribolaceous formulation, the one of mechanism snack is it increase the pH around the peptide and in microenvironment, that is called the microenvironment pH. Around the pH is peptide is called microenvironment pH. So they increase the microenvironment pH, which prevent the activity of pepsin and prevent the harsh environment degradation in stomach. And ribulus is delivered from the stomach. So in this picture, the another mechanism of snack we will discuss. So there is a four pictures. One is A, B, C and D, which explaining the mechanism, exact mechanism of the peptide, sorry, snack. So this is the snack molecule, this is the oligomeric form of peptide, and this is the monomeric form of peptide. So one of the mechanism that the salt caprosate sodium convert the oligomer into the monomer. So how, why monomer is important? So the penetration of oligomer form, it is much difficult to the as compared to the monomeric form. And the another linker lipid is attached to the peptide. The lipid moiety is also attached to the peptide. So both this will help to the penetration of the peptide into the plasma membrane, and this picture is depicted the plasma membrane. Now the big diagram demonstrated how it increases. So the linker wraps around the helix. So first this linker moiety which wraps around the helix, due to the wraps around the helix, it will tilt in some angle, and here is showing that the snake moiety is entering into the membrane and increasing the pore size as depicted into the diagram D. So now the C diagram. So now in C diagram mentioning that lipid dissociate from the peptide and enter into the membrane. So first this lipid will dissociate and this snake moiety increase the pore size. After increasing the pore size, this lipid will dissociate from the peptide and the linker will tightly attach to the peptide. So it attract the peptide. This linker, due to the tightly attached linker moiety, it will cause the entering of membrane of peptide into the membrane. So in diagram 4 depicted that the wrapped linker locks the peptide to the lipid. So because this linker is tightly attached to the peptide, due to this phenomena, it will help into enter into the membrane. Now helix follows into the fluidized membrane, then peptide tilts and straight into the membrane. So the size is increased 20 angstrom. Due to the this increased pore size will help to the increase into the penetration. So this is the mechanism showed by the salt caprosate sodium. The three mechanisms. First is increase the pH. Second is convert into the oligomer into the monomer, and this monomer will enter into the plasma membrane. And the third one is to the increase the pore size. And this due to the these three mechanism of one excipient increase the role of this snake into the further discoveries through this salt caprosate sodium. This phenomena is shown into the ribolaceous formulation. So in this slide, we are focusing on the another permeation enhancer, which is mostly used by the, sorry, this is, okay. So in this slide, we are focusing on the sodium caprate, which is mostly used by the pharmaceutical industries as permeation enhancer or absorption enhancer. There are multiple types of medium chain fatty acid like caprylate, which is carbon chain is 8 carbon, and this in caprate the carbon chain is 10 carbon, and in another one is laureate. So most of uses sodium caprate as compared to the other, because its permeation enhancement activity is more than the other permeation enhancers in this class. So here is the explaining the mechanism of action of sodium caprate. One is the paracellular permeability enhancement mechanism, and another is the transcellular permeability enhancement mechanism. As we know, in case of sodium capillate, the transcellular permeability enhancement effect is less as compared to the paracellular, and its paracellular permeation enhancement effect showing by the affecting the cell signaling pathway is like depicted in this picture. So we are understanding this in less time, because this is not our main concern to understanding the mechanism of that sodium caprate. So first it activate the PLC, and this activate the PIP2 and DAG, and this activate the convert into the IP3. This IP3 increase the calcium by activating the sarcoplasmic reticulum, and this causing the phosphorylation of myosin light chain kinase, and this phosphorylation weakens this bonds between the trijunctional proteins like occludin, claudin, and the another mechanism is that they causing the mitochondrial swelling. This cause the ATP depletion. This also weakens that bond. This is the major mechanism action shows by the sodium caprate, and this sodium caprate used in the many formulations, many marketed formulations, which is sorry, not marketed formulation, which are under the some clinical trials going on that by using this sodium caprate. So in this slide we are focusing on the hydrogel formulation, which is created from the recent uploaded patent, and I am unable to mention the name of the patent due to the some reasons, but we are understanding the whole mechanism in the how they form, how they deliver that formulation, and how they will deliver that formulation. So in this picture is showing that the first is the capsule formulation, which is entry coated, then it release the formulation into the standard. This is the release formulation, and this is the swelled hydrogel picture, and this is the release peptide, and this is picture is depicted and demonstrated that the trypsin and chymotrypsin, and this is a degradation product. As this mentioned, this star mark shows that it is a sodium caprate, and this is the degradation product. Sorry, this is the soybean trypsin inhibitor. So now first we understand how they shows the mechanism and how the peptide delivered by this formulation. So this is the entry coated capsule which release into the formulation into the intestine. Why it is not released into the stomach? Because it protected by the entry coating. And when it release into the intestine, there are multiple excipients are around this peptide. One is the soybean trypsin inhibitor, one is the sodium caprate, one is the or hydrogel. And when this hydrogel come into the contact of the intestinal fluid, it will swell. And we took an anionic hydrogel. Why we took anionic hydrogel? The reason is that we want to repel our peptide from the hydrogel, and the reason is behind that we will discuss further. The fixed negative charge which possessed by the hydrogel we called dominant potential, and the other thing is that the peptide repel from the hydrogel. Why peptide repel from hydrogel? Reason is that the peptide possesses negative charge into the intestine. The negative charge according to the pH, because it's pi, it's pi less than that the pH. When the pi isoelectric point, pi depicts the isoelectric point. The isoelectric point is less than the pH, then it possesses the negative charge because it's the protein and peptide in nature. So due to the negative charge, it will repel from the hydrogel. So why it is necessary? So here we discuss that why it is necessary, because when we see the trypsin and chymotrypsin, this responsible for the degradation of the peptide into the intestine, and they possess positive charge into the intestine because their pi isoelectric points is more than that the pH, and the isoelectric point more than that the pH, they possess positive charge, and they attracted by this hydrogel and decrease the concentration near the peptide. of that enzymes. Okay, so and another degradation, prevention of degradation will help by the our soybean trypsin inhibitor. It will degrade that peptide and more decrease the concentration near the peptide, and the sodium caprate will help cross the biological membrane by opening the tight junction. So how the hydrogel formulation shows the increase the permeation, and it is undergoing the trials, an in vitro trial and maybe some preclinical trial. So this is the recent uploaded patent shows the hydrogel formulation. So now here we are the ending of our presentation. So in conclusion, we are conclude that oral delivery of peptide and remains challenging due to the enzymatic degradation, as we already discussed in our challenges, low permeability and instability in the GI tract due to the physicochemical properties, degradation by enzymes, and other issues also we already discussed. Now second point is that advanced strategies like combinational strategies, using of ionic liquids, the chemical modification, lipidation, pegylation, or use of snack in marketed formulation also we discuss in the riblases. Then the fatty acids significantly improve the stability and absorption. Then the combined permeation enhancers, as we know, combining of permeation enhancers and innovation formulation technologies offer the promising path to achieving the effective oral peptide therapeutics. So this is our conclusion. Now next is our thank you. So thank you for watching.